Hello everyone, I'm Rama Anisha, third year computer science student at Bennett University. Bennett University is a world-class university with innovative education and fueled by many global academic partnerships. Bennett University comes under the Times of India group, which makes education more than just a learning experience. At Bennett, the student can get diverse contacts from different streams and increase connections, get in-depth knowledge from its vast knowledge base, growth in skills and innovations, and a student can build the best career. Bennett University pushes students to its limits, innovation-first campus. It helps us in real-world problem solving, and it's a student-centric education. So let's now talk about an interesting and groundbreaking stream in computational science field, DNA computing. What is DNA computing? A DNA computing is an emerging branch of computing which uses DNA, biochemistry, and molecular biology. As quoted by Bill Gates, DNA is like a computer program but far more advanced than any software ever created. So now we are going to see what DNA computing is, its pros and cons, its applications in real world, and I'll also provide the references which I have used to make this presentation. Let's begin with what is a DNA computing. We all know that DNA stands for deoxyribonucleic acid, but what does it represent? It represents a genetic blueprint of living creatures. It contains instructions. Every cell in human body has complete set of DNA. DNA is unique for each and every individual. What is so unique about DNA anyway? It has extremely dense information storage. It has enormous parallelism and has extraordinary energy efficiency. DNA computing is an area of natural computing based on the concept of performing logical and arithmetic operations using molecular properties of DNA by replacing traditional carbon or silicon chips with biochips. Let's talk about its history. DNA computers are invented by Leonard M. Alderman. His article in 1994 issue of a journal Science outlined how to use DNA to solve a well-known problem of mathematics, that is directed Hamilton part problem. Silicon-based microprocessor will not be made any more powerful in the foreseeable future. DNA computing is a new technology that uses DNA molecules to create computers that are quicker than the most powerful human-made computers. By evaluating the patterns of mixtures and the growth of the molecules, the DNA computers employ the chemical properties of the molecules. The input and the output of DNA are both strands. This computing is also called as molecular computing. These operations are carried out in the form of sequence of biochemical procedures. They are also used for synchronizing, modifying, and extracting. Now, let us look at the differences between DNA and conventional computers. Firstly, storage type. Storage is one of the most important aspects of a computer. There are different ways to store data and different types of storage, but it basically boils down to semiconductors. Conventional computers use semiconductors, but DNA computers use nucleic acid, which can store very large amount of data, more than 10 times the storage capacity of a semiconductor. Now the memory. Memory plays a vital role in functioning of a computer. Due to the use of nucleic acid as a basic component, DNA computers tend to have very high memory, whereas conventional computers have high memory. Conventional computers have logical operators, that is OR, AND, XOR, XNOR, NOR, logical operators. Whereas DNA computers use very clever techniques to mirror these operations using biochemicals. These are known as biochemical operators. Operations. Conventional computers run operations in a sequential manner, that is, one task after another. But DNA computers can run tasks parallelly. Hence, DNA computers can handle large data. Conventional computers run on silicon, which is very good semiconductor. Hence, the electrons flow freely and quickly. But DNA computers run on biochemicals, in which electrons does not flow freely. Hence, conventional computers are very fast compared to DNA computers. Conventional computers are deterministic, which means when given certain input, we know that we would get the exact same output every time. But DNA computers are stochastic, that is, the output varies even if same input is given each time. There is some randomness in stochastic process.
Now let's look at the benefits and drawbacks of DNA computers. They have the advantage of being very good at solving non-deterministic polynomial problems that are NP problems we have learned in the essay. In terms of size and power consumption, it is really small. Massive amounts of data can be stored in small amount of space. The disadvantages are it requires constant supply of proteins and enzymes, which are very expensive. And DNA computers need human intervention. Now let's talk about the advantages of DNA computing. There are many advantages of using DNA instead of silicon chips. Some examples are parallelism, gigantic memory, low power deprivation, suitable for combinatory problems such as NP problems, clean, cheap and available because they don't use any harmful chemicals and they're naturally available in the nature.
Let's discuss few existing problems in DNA computing. DNA computing is mostly theoretical and have not discovered any killer app. DNA can solve most problems that a regular computer can, though not always more efficiently. Now let's answer a few common questions. Is DNA digital? DNA contains two kinds of digital information. Genes that encode proteins, which are the molecular machine of life, and gene regulatory network that regulate how the genes behave. Is DNA computing the future? DNA computers will be thousands of times smaller than a silicon-based computers and tens of thousands of times more powerful. 1 pound of DNA can store more information than all the electrical gadgets ever developed. DNA computers as small as a water droplet will have more computational capability than today's most powerful supercomputers. Let's talk about its applications. DNA computing has many real-world applications such as scheduling, security, cryptography, Turing machine, and clustering. First, scheduling. The job scheduling problem is solved using a DNA computing-based method. To describe the concept, the working operations are 6 tasks that imitate the way used to solve the Hamilton path problem. Early in the millennium, DNA scheduling algorithms were employed for elevator schedule system and the reorganization of the flexible manufacturing system. Number 2. Clustering. Clustering is concerned with determining highly relevant correlations in a large collection of data by constructing a structure with the help of various concepts and methods. Edges and vertices are assigned using DNA strands in DNA-based clustering. To increase quality, iterative calculations are done on each created cluster. The strategy is very useful when dealing with vast amount of heterogeneous data with an undetermined number of clusters. It aids in lowering temporal complexity due to DNA's strong parallelism. Number 3. Security. The ability to store 108 terabytes of data in 1 gram of DNA has resulted on the possibilities of holding a massive one-time pad. Another example is DNA steganography, which uses a revolutionary approach to conceal messages in a microdot. Instead of standard binary encoding, each letter was represented by 3 chemical bases, such as CGA for the letter A. These messages are then encoded into DNA sequences and disguised by mixing them with a vast amount of random human DNA in a tube. This resulted in production of microdots, which the receiver decoded using proper short sequence with complementary bases. Number 4. Cryptography. Currently, researchers have devised a method for DNA computers to crack messages encrypted with the Data Encryption Standard (DES). Although this problem has already been solved using traditional techniques in considerably less time than the DNA method offers, the DNA models are much more versatile, potent, and cost-efficient. Number 5. Turing machine. The DNA computer is a virtual Turing machine. Several theoretical methods for building a universal DNA computer has been devised, and mathematical proofs have demonstrated that some DNA computing models are at least equivalent to conventional Turing machine. Some institutes created a programmable and autonomous computer machine in 2001 using DNA strands as input and transition rules. Now let's learn about the advancements in DNA computing. The most significant advancement in the field were made in the years 2013, 2017, and 2021. In 2013, a team of Stanford University bioengineers led by Drew Eddy developed the biological counterpart of a transistor, known as a transcriptor. The invention was the third and the final component required to construct a fully operating computer. In 2017, a team led by colleagues from Arizona State University's Biodesign Institute and Harvard's Wyss Institute for Biologically Inspired Engineering created a biological computer within E. coli that responded to a dozen inputs. In 2021, a team led by biophysicist Sangram Bhag completed a work with E. coli to tackle 2x2 maze puzzles in order to investigate the notion of distributed computing among cells.
In future, as technology becomes more refined, more efficient algorithms may be discovered. The University of Wisconsin is experimenting with chip-based DNA computers. DNA manipulation technology has rapidly improved in recent years, and the future advances may make DNA computers more efficient and useful. So to sum it up, the key learnings we can infer from this information is that we have to overcome many issues to produce a useful DNA computer. It will not replace the current computers because it's application specific, but has a potential to replace the high-end research-oriented computers in the future. Here are the references which helped me in building this presentation. Please check this out for further information. Thank you everyone. Have a great day.